Good afternoon and welcome to today's webinar. Today we're coming to you from the University of Ghana in Accra, Ghana, West Africa, and specifically from two units within the university, the West African Center for Cell Biology of Infectious Pathogens, for which I am the director, and then the Ngoi Memorial Institute for Medical Research, which is also within the University of Ghana. So since the pandemic hit our country, we as scientists at the University of Ghana, we've been doing our part to contribute to the response and to provide useful data for the Ministries of Health and the Ghana Health Service to guide the response strategy. One of the things that we have been doing is sequencing the viral genomes. So today we're going to talk a little bit about some of the sequences that we have done so far and what we can learn from them. Today we have two presenters. We have Joyce Ngoi, who is our next generation sequencing manager, and she is the one who is responsible for actually getting the genome sequences done in the lab. And then we have Dr. Peter Quashie, who is a senior research fellow and a virologist, and he would present the results and whatever we can gain from the results so far. So we'll start with Joyce. Joyce will take us a little bit, behind the screen and tell us how the sequences were generated. This, we think, would be useful for colleagues in other African countries who are trying to sequence and who may have difficulties in getting good data. So we'll share with you how we managed to get this done successfully, and we'll be happy to take any questions about how this can work and in terms of troubleshooting any challenges that others may have. So Joyce, we'll start with you.
Hello, everyone, and thank you very much for the introduction, and many thanks to the IURS for giving us the opportunity to share the work we've been doing in Ghana with regards to SARS-CoV-2. So I'll take you through the sequencing workflow, and my colleague Peter will give you a summary of what we've found so far. Like Prof has mentioned, this work is a collaboration between WorkWith and UBC Memorial Institute. So as we are all aware, the SARS-CoV-2 virus emerged from Wuhan, China in December 2019. And even with the efforts by the government to lock down the city, the virus managed to spread all over the world, resulting to many cases of the disease across the world. So in Ghana, the first case was reported on March 12 of 2020, and around the same time, the global count was around 160,000 cases. And then 2 months later, we started seeing an exponential growth in the number of cases reported. And at this point in Ghana, we reported over 5,000 cases, and an estimated 4 million cases across the world was reported. As we speak now, there are over 41,000 cases reported in Ghana and an estimated over 20 million cases worldwide. So from the figures, you can see that the virus has managed to spread very rapidly across the world. And the way that viruses manage to do this is viruses usually replicate rapidly and multiple times within a host. And the replication mechanism of viruses, especially RNA viruses, is very error-prone. And this leads to accumulation of mutations in their genome over time. Now, these mutations are important to the viruses. They help the virus to adapt to the host, but we can also use the mutations as markers of transmission. And this is where sequencing comes in. So we sequence, by doing whole genome sequencing, we can identify these mutations in the viral genome. And then from there, it can give us an insight on how the virus is evolving over time, and then also use this information to track the transmission chain within the community. So why sequence? So the first reason why we wanted to sequence is to understand where the virus was being imported from. So just to know the source of or the origin of the infection. And secondly, to see and confirm the evidence of local transmission, and also to understand the transmission dynamics of the virus in the country. So the last question we wanted to answer is to see how the virus is evolving in our population, and this would do by identifying any lineages or clades that are circulating within the country. So we managed to do our sequencing in 2 batches, and the first batch of sequencing was from samples collected from earlier on at the beginning of the epidemic. So we managed to do this within the first 2-3 weeks after the first case was reported. And then the second batch of sequencing was done from samples collected around 2 to 3 months after the first case was reported. And this is the time we were seeing an exponential increase in the number of cases reported. I'll talk in brief about the sequencing workflow. And just to mention, one of the main aims was to establish and optimize a standardized protocol that can be used in our lab as we plan to sequence throughout the epidemic. So once the sample is received in the testing lab, RNA was extracted, and then the extracted RNA is subjected to qPCR to confirm the presence or absence of the virus. And at this point, this is the point where we select the samples that are suitable for sequencing. So for the first batch of samples, we, like I mentioned, we sequence samples from collected from the beginning of the epidemic, and most of the samples collected around that time from these patients, the patients were asymptomatic, and the viral loads were a bit low, as you can see from the CT values. So most of the samples we had a CT value of between 22 to 35, and we realized that we had a lot of challenges working with samples that had very high CT, hence losing a bit of the sample. So we started with 20 samples and managed to sequence 15 samples, 5 of which failed the QC cutoffs that were put. So for the second batch, we decided to start working with only samples that had a CT value of 25 and below. So we had 36 samples for this batch, and we only managed to lose 4 samples from this batch that did not pass the QC checks. So the third step is, after we identify the samples that are suitable for sequencing, then we go back to the stored sample and re-extract the RNA. And this is because we noticed that if we go with the standard protocol that was being used in the lab, we don't get enough material for sequencing. So we sought to amend our protocols a bit so that we can increase the yield of the RNA that we wanted to use for sequencing. And then after that, we QC the RNA just to make sure that we have enough quantities, and also we check the integrity of the RNA just to make sure that we have a good quality RNA that we can use for sequencing. Then after that, we prepare the cDNA for the next step. So for this step, this step involves target enrichment, and which is the virus in this case. So the reason we want to do virus enrichment is because we want to increase the amount of the viral material in the RNA. And this is because once you have your total RNA extracted, you not only have the viral RNA in the sample, you have human RNA, which forms the bulk of the sample. You have bacterial RNA as you're collecting the sample from the upper respiratory tract, which is colonized by a lot of resident bacteria. So you have a lot of RNA from the bacteria in the sample, and probably other respiratory viruses in the sample. So you want to enrich for the viral material to generate enough material, sufficient material for sequencing. And this in turn will help in reducing the sequencing background, as all the reads that you're going to get from the sequencing are going to be on the target. So for this process, we opted to use the ATIC protocol. And the ATIC protocol uses a multiplex tiling PCR approach with approximately around 198 primers for version 1, and an updated version of these primers has over 200 primers. So these primers allow you to generate short genome-wide overlapping amplicons in enough quantities to form a tiling path across the entire 30 kb genome. So the PCR occurs in two different reactions, with each reaction having a set number of primers in each reaction. So each primer pool will have primers designated in that primer in such a way that you have alternate primers targeting a particular region of the genome in that particular primer pool. The reason is because you don't want to have neighboring primers in the same reaction, as you end up preferentially amplifying short overlapping amplicons. So as illustrated in figure B, that's how you assign the primers into the primer pool. So you have, for example, if it's the V1 primer, you have 98 primers in one pool and another 98 primers in another primer pool. And once the reaction, the PCR reaction is over, then you pull all the products from both PCR reactions to one pool, and then this pool has all the amplicons representing the entire 30 kb genome of the virus. So in this, at this point, you only have the viral material in that sample. So, and of course, you want to QC your assay just to make sure that you have the 400 base pair product in your sample, because the amplicons that you are getting were around 400 base pairs. And some of the cases we managed to see that some of the samples are not amplifying, so those ones will be left out of the assay. So the next step was the library construction, and it's at this point that we managed to adapt the ATIC protocol for use with the MiSeq in our lab. The ATIC protocol is designed and optimized to work with the Oxford Nanopore platform. So at this point in our lab, we sought to adapt the protocol to work well with the MiSeq. So we optimized the protocol. We chose the Nextera XT Illumina kit, which basically uses transposons to fragment the amplicons, and at the same time tag the amplicons with adapters, Illumina adapters. And then immediately after that, we barcode each sample with unique sequences or unique indexes, so that when we pull them together into the final tube for sequencing, you'll be able to sequence as many samples as possible. Then, of course, the next step is to QC the library. So you want to be sure that you have the properly ligated library, and we do this by doing a fragment analysis. The fragment analysis is done using the analyzer, and at this point, you want to check the size distribution of your library to make sure that you're getting the correct size based on the size of the amplicon you started with. And then next is to quantify the library. So we quantified using qubit, and once you have enough material to go ahead with it for sequencing, then we normalize all the libraries at this point. So all the samples that we prepare the libraries for. So we normalize them to an equimolar amount and then pull them into one tube. So from this point, we'll be working with a single sample consisting of multiple samples inside it. So the number of samples we pulled for us was we were targeting to have a good coverage of the genome after sequencing. So at this point, it's very important to make sure that you don't pull too many samples and then your coverage will be low. So we were targeting at least 50x, but we ended up getting a lot more than even the 50x coverage, because you want to be sure that once you start doing your variant calling, you're very confident. And then the final step is to dilute down the library so that you can have enough signal for sequencing, and then we spike it with a sequencing control and we sequence it on the MiSeq using Illumina version 2 kit for 500 cycles. I'll hand over this point to my colleague Peter to give us a summary of the findings. Thank you.
Thanks, Liz. So moving on from once we have the sequences, we do a bit more QC. First to check the quality of the FASTQ sequences, and then to confirm that they map to the SARS-CoV-2 reference genome from Wuhan. Also, then phylogenetic analysis was carried out to identify the major clades. And once we had that, we used the sequences, the clades, as well as the mutational patterns that we observed to infer transmission patterns within our sequences and within the country. So this is a busy slide which basically goes from A to Z, and we are here at the Illumina sequencing. We have the FASTQ files, which we then subject to sequence analysis using FastQC, which just basically checks to make sure that there's a good coverage of the sequences. The amplicons are all a decent size; they're not too small, and the integrity of assignments at each base is correct. Once that has happened, we trim the sequences using trim Galore. What that does is it trims off the adapter sequences. So in this case, the Nextera adapter sequences. And once those sequences are trimmed, you should have all the remaining sequences should have enough read depth, and they should be of appropriate length. They shouldn't be too short, otherwise it gets rejected. Then after trim Galore, you go back to another QC to make sure that the trim Galore did not decrease the integrity of the data. Once you have that, and then you have your Wuhan reference sequence, you use a Burrow Wheeler tool to align the sequences and to come up with the to come up with the coverage, and then do phylogenetic analysis. And this is a simplified version of that, just showing you that we had started off with 51 samples. After sequencing and QC, we left out 3 samples, so that gave us 48. And then after doing the Burrow Wheeler alignment, we left out actually 2 samples. So in the end, we ended up having 46 samples, which have all been submitted to GenBank. And some of these, the first batch have been submitted to GISAID, and the second batch is underway for submission. And of these sequences, we were able to assign clades to 43 of them. So those were included in phylogenetic analysis that I'll go into henceforth. This is just, again, going back to the quality of the reads that we had. This is a subset of the data from the first batch. You can see that this was using the version 1 Arctic primers. You still had about almost 100% of the reads mapping to the genome, and almost 100% of them passing the quality control with a high specificity. And this is this is very busy, I know. This is the entire 46 sequences. What you see in black here are all the portions that were not able to align with the portions which had high ambiguity. And again, there was an improvement from version 1 to version 3, where we had an improvement of minimum coverage from 91% to actually 96%, which means that in the first 15 sequences that we used the version 1 primers, we had one sequence which had only 92% coverage, and with the version 3 we ended up having only one which had 96% coverage. So 38 sequences had very low N. So N just means where there's not enough data to assign a base. So only 2.5% of 38 sequences had N's in only 2.5% of the positions. 45 had N's in less than 6%, and only one I mentioned had N in 9% of the positions. So the parts in green are also showing the mutations in our sequences relative to the Wuhan strain. And this is just a snapshot of GenBank showing that the sequences are there and they're live. And going back to the initial 15 samples that we sequenced, we can see that Ghana is here in orange. We can see that our initial samples, including the ones which were identified as imported and not imported, imported and including the ones identified as coming from people who had no travel history, they all clustered around sequences which were mostly from Europe and the US. There was only one sequence that clustered with Indian sequences, and that's because that was from an individual who arrived from India. Again, going back to that sequence, our 5 imported strains, our 5 imported viruses, which we included in the sequencing, all link back to the countries of origin. That was the US. There was one that the individual traveled from the US through Europe, UAE, and then to Ghana. And that sequence strongly clusters around sequences from the US, showing that that particular individual, the virus came from the US and it was imported directly into Ghana. Even though we have some sort of diversity in our sequences, they don't, apart from at the major reported mutational loci, they are quite different between Ghana and Africa, and that's because what has been reported is that most of our strains are imported strains, and there's yet to be significant evidence of country-to-country spread. Going into the data, I know this is the text is a little small, but this is I'll show this again in trees. This is basically showing the clade assignment, and currently there's 3 main methods of assigning clades to SARS-CoV-2 sequences. There's a legacy clades, which was what was used very early on in the outbreak and the pandemic, which is, in this case, we have a lot of B4, clade B4 sequences. And then if you use GISA clades assignments, we have a lot of S, and that S is basically refers to non-synonymous mutation in open reading frame 8, which is L84S. So all sequences which tend to have that non-synonymous mutation would be classified as GISA clade S. There's another assignment which I think is gaining momentum, and that's what is primarily used on next strain. which is 19. So in this case, 19B sequences will probably be 19B. And that's based on the year of detection of that main mutation, and which has spread significantly so as to be assigned to a clade. So in this case, it was in 2019, and it was the second such mutation to be assigned, so 19B. And what's interesting about SARS-CoV-2 is, as Joyce mentioned, normally RNA viruses have very high rates of mutations, high rates of mutagenesis, and very rapid evolution. What we've seen with this virus is that you have long periods of stability. So the other half of our sequences all tend to be what we call the G, G1, G614G variants. So these are all classified as G8 clades, G, GH, or GR, depending on what subsequent mutations and non-similar mutations come up from there. And they are also referred to as legacy clade A2A. I'm mentioning all of this so that people can sort of correlate the two. And we also have the pampering assignments here. So what you can see immediately is that, looking here, we have greater than 50% of all our sequences are starting to have the G614, the D614G non-synonymous mutation. So all of them are expressing this variant. And we actually have a huge representation in our second batch of the clade GR. So that appears to be the fastest detected sequence in our second batch of sequences. This is another way of showing the data. As I mentioned, we had it detected before in our February, March sequences, but we detected it in a significant proportion of our samples from individuals with no history of travel. So from the two batches, we have 19B, which is the same as before, or the same as just a kid S, talking around about half of the sequences. Then we have from this side, all of these are D614 G variants clades. And this is a time plot showing when the sequences were taken for the different sequences. And what we can see looking at the two batches. I touched on this earlier, but there was a lot of diversity in the first sequences there was a lot of diversity in our first batch, as in we had 5 clades being represented. So we had 19A, which I haven't talked too much about because we saw significant levels of deletions in those samples, and then we also saw 19B, which is the one which we didn't detect in imported cases. We saw also, even in the first batch of sequences, the G614 is quite well represented. But what's striking is that in our second set of sequences, we see there becomes a shift to go towards the S sequences. Okay. So there's a shift towards the S sequences in the second batch of samples, and it almost ends up being that about 90% of our sequences are now either exhibiting the L84S substitution or the G614G substitution. So basically, the take-home message from this slide is that our 46 genes that we sequenced in Ghana exhibit a lower than expected level of evolution. There could be a number of reasons for this, including maybe a lower level of transmission. And digging into this, going beyond just the non-synonymous mutations and looking at the synonymous mutations as well, we're able to look and go in and see if any of these sequences are related to each other and whether we can see any evidence of one virus being transmitted and being shared by multiple people. So I'll focus first on the S-clade variants. And then in this case too, we were able to identify viral phenotypes which we didn't sequence but must exist for them to facilitate some of the sequences that we see. An example is this phenotype here. So when it's encompassed by a red box, this is one that we suspect has the ability to form a cluster and transmit, because we have this mutation which we couldn't find in, at least in our hands, we couldn't find in any of the reports worldwide. So when we can't find it in a report and we see it in more than one sequence, then we take that as a local evolution. So we had this backbone virus, which we detected in you can see the full screen. Okay, so this one basically, based on the sequence alignment and relation, gave rise to sample 3 virus from 15 okay. So then there was evidence that these 4 individuals got their viruses from somebody who had this genotype, and that is evidence that this genotype is likely circulating within Ghana. Of course, this S mutation L5F has been reported elsewhere, but we couldn't find any of the other accompanying mutations. And this has also been reported as a locally evolved mutation in India. So it's probably a locus which tends to spontaneously mutate and not necessarily transmit. We also have these cluster of genomes here, where this particular specimen 45, there's clear evidence that this was transmitted to these 2 individuals. And we can also tell this by looking at the metadata underlying these sequences and knowing where the individuals live. So these 3 sequences were all isolated from individuals who live in a suburb of Accra called Ayawaso. And that is known to be an epicenter of the local outbreak. And then some of these sequences are also from Ayawaso. But we have some which are emanating from the central region and then the western region, showing that you have transmission from one region to the other. What we're seeing from the S-clade viruses is that we have we have sequences which are linked to each other from both Ayawaso, that's within the suburb of Accra, Tema, which is on the other side of the city, and they're linked to samples which are within the western region. I'll show a map shortly which will sort of indicate the transmissions that we're talking about here. But what we found was quite interesting here was that we had 3 genotypes which we didn't detect in our sequences, but which must be circulating and which must be forming transmissions, because based on, I'll start from here, we have this sample 47 and 36. They both share the mutation in spike I805M, and then they have additional mutations. However, they also share this 3 sets of mutations, A26, B96, 92K, 365, which is the same mutation as this virus, this isolate here. So these 3 must have all evolved from this locally circulating virus. And I will say again that when they have red around them, we think that they are transmissible. And these must also evolve from this strain. So I'll go on to the D614G variants, and which I said were roughly 50% or more of our sequences. When the sample number is in yellow, that's an imported strain, so that's not one that we have any evidence of spreading locally. This is isolates that we got directly from people who had just arrived in the country within a week or so. So of note, there was a cluster of sequences which seemed to be transmitted from military personnel who were based in Accra, central region, and the western region, and again linked back to Ayawaso. I'll just show a map of Ghana to give a better appreciation. This is Accra here, which is... Oh, they can't see the mouse. Okay. So in Accra, which is the capital, has the most number of cases within the country. And then this is this, right next to Accra, where you see Cape Coast is the central region. And the next region over is called the western region. And so we were able to show that we had viruses, isolates from the western region and central region, which were related to isolates in Accra. And what's interesting is that while we're able to show that we have a high level of D614G variants in the country, our cumulative number of cases as at today is just 45, well, it's 45,505 with 261 deaths and a case fatality ratio of 0.5. So taking in mind that these sequences we did were from the end of May and beginning of June, and that was at the beginning of our exponential growth, which is likely linked to the D614G variant. What we show here doesn't seem to support a role of that variant in deaths. But looking at the distribution of deaths, even though Accra has the highest number of cases, it doesn't have the highest number of deaths. So moving forward, that's something that we have to look at. However, our death breakdown in the country is not very different from what we've seen in other places around the world. So we'll summarize by saying that we've demonstrated an ability here at WACDIP and at Ngoi to routinely perform high quality next generation sequencing with, we have in-house expertise for phylogenetic analysis. And what we noticed from our cases as reported elsewhere, initial cases were mostly imported from Europe and the US, not Asia so far. And these two clades, S and G-linked clades, make up 90% of our viruses that we've sequenced so far. There's evidence of transmissions from Greater Accra through Central to Western region, and this includes military personnel and high prevalence of D614G does not support a role in high mortality. Moving forward, we're looking at sequencing 200 to 300 sequences to properly track the virus around the country. This is using Nanopore sequences. So the first 15 also sequenced by Nanopore and showing that we can get comparable data between MiSeq and Nanopore. And then linking up better with the GHS and clinicians would allow us to tie our sequences much better in terms with the outcomes of those patients and disease severity. We've started doing both in silico and in-lab analysis of some of the transmissible variants that we've had. So I'd like to thank, we would like to thank the entire team and our Collins here did most of the phylogenetics that I presented here, and virology did a lot of the extractions and we're doing the testing. Yes, and I'd like to thank IUIS for this opportunity.
Okay, all right. So, thank you very much for your patience. We apologize for the technical challenges that we had with Peter's computer. So we'll take questions for about the next 10-15 minutes, and then we can wrap it up. So I was looking at some of the questions. I think there was a question about whether saliva could be used. I think, Joyce, you want to answer that? So maybe you can tell about what specimen we used here and what do you think about saliva?
So we used, for these two batches, we used nasopharyngeal and oropharyngeal swabs, but saliva could as well be used. The viral load might be a bit lower, but it's still, we can still use it and extract RNA for sequencing from saliva.
Okay. Good, thank you. There was a question about whether you saw new mutations when you compared the first sequencing to the second sequencing. So, Peter, yeah, I think we talked about that, but because the sound was bad, I think we need to recap some of this.
Yeah, so we did see some new mutations between the first and the second. And I showed that on those two slides. What we did see in the second batch was that there were a number of mutations that seem to have arisen from an undetected strain. So, I mean, I think these slides will be available. So if you look at that, you can see some of those mutations. And these mutations that we've identified in Ghana, so far I haven't seen them on any reports around the world. And those are all shown in blue in the slides, with the exception of the the 5L. Yes.
Okay.
L5X.
There was also a question about the D614G mutation. I think you talked about that also, but maybe you can recap for those who couldn't hear you because of the sound.
Okay. So what we've seen is that the D614G mutation is becoming one of the predominant clades in Ghana, and interestingly, all the imported sequences that we sequenced were D614G containing.
Okay. Let's see. Were there sequences that were specific to Ghana? Somebody asked that question. That were different from seen elsewhere in Africa.
So in the 19A clades, we have a high level of deletions, and those didn't quite align as well, and some of them we omitted from the analysis. And I've seen some reports from Nigeria where there are some sequences which don't quite align, and those have been purported to be locally evolved, but that's something that we probably need to look a little bit more at.
Okay. There was also a question about any association with disease severity, which is, again, I think something you mentioned that we don't have sufficient clinical data, but you can explain.
Yes, there's only one of our most evolved strain, one of our most evolved isolates. That was actually an imported isolate. Was the patient was in ICU under ventilation, and that individual had mutations both in spike and nucleocapsid. Unfortunately, we haven't been able to tell what the outcome of that disease was. However, we know that globally, patients under ventilation have a high chance of dying, and I think that number is actually higher in Ghana. I mean, they have a 50% chance of dying, and that's higher in Ghana. Typically, our patients don't tend to have severe disease, and once they go to severe disease, prognosis is very poor.
Okay, so maybe it has more to do with underlying conditions and the time at which they report to the hospital more than the viral sequence, you would say?
Yes, so it looks like, according to the Ghana Health Service, 75% of deaths are due to people with underlying conditions. And as I showed, our death profile in relation to age is not that different from the rest of the world. However, our case fatality rate is, if you factor in the death profile, is still quite remarkable.
Remarkable in a good way?
In a good way, yes.
Okay. All right. Related to that, there's a question about whether you think the mutations you are seeing would impact on the efficacy of vaccines that are under development. So in other words, where you are seeing the mutations, most of the vaccines are probably targeting the spike. So
yeah, so there are a number of spike mutations. I mentioned that individual had 2 spike mutations. But if you actually look at the structure, where those mutations were located were on the external, they would be interfacing with the membrane, not necessarily with the host. So currently it's unclear how they would affect the vaccines that are in development. We haven't identified anything that would be within the receptor interaction interface, and it's actually, I think these probably are more affecting other cellular interactions rather than a direct inhibition of
the binding.
Yeah.
Okay. I think we've covered the range of the questions. Was your genetic tree able to pinpoint the exact time the virus came into Ghana?
So I wouldn't say exact time, but there's an analysis that I didn't show which seemed to suggest early March, late February. So that seems to, based on the number of mutations that we observed in our sequences. The caveat there is that because we didn't detect the B4 clade in our imported sequences, we can't be quite clear when that came in, but it's likely around the same time too.
Did you include samples from asymptomatic persons and reinfected persons? That's a good question.
So first batch were mostly asymptomatic. I think there were a few people who were sick, but not seriously, with the exception of that patient who was under ventilation. The second batch, I think, was even more asymptomatic patients than the first batch. But there were some symptomatic. So far in Ghana, most of our cases are either pre-symptomatic or asymptomatic or very mildly symptomatic. So we haven't, as yet, been able to do that proper analysis. So we're hoping that if we can get a full metadata for some of these samples, we can do this better.
Okay, there's a technical question, Joyce. How many samples are you able to sequence on one nanopore flow cell?
So this depends on the kind of application you're using. So if you're doing, you're enriching for the virus using the protocol I use, the active protocol, then with that one, you can sequence as many as 24 samples within one flow cell. But if you're doing you want to do metagenomic analysis, then you probably have to do less than that, because like I mentioned earlier, your RNA will have more than just the viral material, and there will be a lot of sequencing background, as the belief that you get at that point will not only be of the virus, there will be from the human RNA and any other bacterial sample. So if you're using a metagenomics approach, then you have to use less samples, probably less than 10. Yeah. If you enrich for the viral material, especially using the PCR method that we talked about, and that one you can do up to 24 samples and get good enough coverage of the genome.
Okay. Okay, good. I think we've covered everything that has been asked. So we would like to thank everybody for, bearing with us despite the technical difficulties. We will continue to update. When we get new data, we'll come back and update everyone about what we're seeing. We're also doing other studies on serology, and we would share with you when we have, more concrete data on those ones. So I'd like to thank Joyce and Peter for their presentation, and we thank IUIS for the opportunity, and we'll see you again soon.